All right, good evening. We welcome back to exam revision 2. We will start from the beginning, so those who were not here the other time can also have a good look at what the requirements are in order to prepare for the exams. So we're still in week 14, and this is Monday, 8th of June. Book. Yep. All right. So for those who haven't downloaded the exam timetable yet, the exams for biochemistry is Thursday, June 25th, and that's the time when you have to write the exams. PET 7 to 1 p.m. and this chemistry is 2.5 hours. Any questions on the time?
Nope.
Yep, everyone's clear on it? All right, so you have 2.5 hours to complete the exams. Welcome back. So that's done. And this is what your exam looks like. This is BIOB final exam, and this is the front cover of your exams. And it shows you the time, the day, and your lecturer's name.
Excuse me, I'm not sure whether I've connected the right way, but I can't see any of your screen. Should I join?
Joe, is anyone there can see this? I've been sharing the screen. Can anyone see the screen?
Yeah, I can see the final exam revision.
Yes.
I can only see myself at the minute, and I've joined without video. Should I?
Yeah, join with video. Possibly.
So it's not doing anything at the minute, so I might have to go out and try to come back.
Okay. Everyone else can see my screen?
Yes, I can.
Okay. All right. We'll give a few minutes. Anyone has any questions? We have a few minutes so to just see if she can come back. Any particular questions you would like to be answered today? That the exams is you're doing that online and you need to download the software. Everyone's done that too?
I haven't done that.
Okay, but you have the information to do that.
Yeah.
Okay. All right. Is Georgia back?
I can hear you, but I just cannot. My screen looks like it's freezing, so I'll have to find a way to come back. So I'm sorry, I don't want to delay you. I will. You can continue, and I'll try and come back.
Okay. All right then. So that's the front cover for the exams, and this is part of the front cover. So what we have here is the duration of the exam, which is 2.5 hours. The total mark you can get is 100, and the weight is 40%. You've already earned 60% already from the quiz 1, which is 20%, quiz 2, 20%, and your written assignment, which is also 20%. So for this exam, part A, if you look on the left side of this, there are 3 parts to this exam. There's part A, which is multiple choice questions. This multiple choice question, there are 36 questions on that. And that is exactly the same as you will have done for your quiz 1 and quiz 2. And most of what the preparation you need for this part of the exam is your tutorial questions. So if you go over your tutorial questions, they are almost exactly the same type questions. And so if those who have done the tutorials and also the review quizzes, you should be able to do well. Most students do very well, and one is 1 mark. One question is 1 mark. So if you get all 36, then you're almost halfway to passing this. The part B is 8 marks. And that is 2 questions. And we'll quickly go through that and show you what that means. Part C, we will do that as well. And the third part is part C, which is the written exam. So this time you are going to type it in. So part A, multiple choice questions. Everyone's clear, and it is beginning from Session 25. Are you able to see now, Georgia?
Hello, yes, I can. Thank you.
Okay, so what we've done so far is just look at the front cover of the exams you would do.
Sure.
So when you look at it, it's 2.5 hours, which you already know on the left side here.
Yes.
And it's 100 marks, 40% weighting, because you've already earned 60% from quiz 1, quiz 2, and your assignment, written assignment. There are 3 parts to these exams. Part A is multiple choice questions, 36. The best way to study for this part is to go through your tutorials. The questions are very similar to the tutorial. So if there's any tutorial question you're not sure of, you can either ask me for a student consultation. I'll give you a call back and we can discuss a particular question or questions. And then there is part B. Part B is only 8 questions, and we'll go through an example of what part B is. And most students get part B. It's straightforward and everyone should get that. For example, it asks you what exam, for instance, if it's on digestion, what carbohydrate digestion enzyme do you find in the mouth? And everyone knows that by now? Does everyone know carbohydrate digestion enzyme in the mouth?
Could you repeat the question?
The carbohydrate digestion enzyme that we have in the mouth.
Alpha amylase.
Nice.
What about
Salivary alpha amylase.
yes? What about the lipid digestion enzymes in the mouth?
Oh, I'm still trying to remember that one.
That would be the lingual lipase. Everyone's clear on that one?
Lipase?
Lingual lipase. And so this you'll have in your tutorials as well. And then the question is, what happens in the small intestines? There are 2 things happen in the small intestine. The first is you break the ester bonds in the lipids, and then afterwards it forms into micelles before it's absorbed into the lacteals. So questions can be asked as which one comes first, the micelles formation or the ester breakup? Which one comes first? And everyone knows that you will have to
Formation.
you have to digest first and then send it off as micelles. Is that okay there?
Yes.
And then the next question is part B. Let's look at the part B before, and then it's the first part of part B is what I've just said, matching. You have 4 questions and you match the correct answer. Typical example is the digestion questions. Or, for instance, you get a question like this, matching glycolysis, and then you say that our glycolysis is B, the breakdown of glucose into pyruvate. Everyone's clear on that one? And that will be 4 questions. Glycogenesis, and if you know what glycogenesis is, it's a definition of glycogenesis, and it's synthesis of glycogen from glucose. You select that.
Yep.
And then glycogenolysis is what? The breakdown.
Breaks pyruvate from glucose.
Yes. And then the last one, anaerobic, is when there is a
Higher oxygen.
without oxygen. So that's type of similar question. So you go and review digestion and review some of the definition terms you have in your tutorials. And that will be 4 marks for you. The other part is this one. And this one is also straightforward. It's trying to find out whether you still remember all the metabolic pathways you have done so far. What's important here is exactly how you have written your assignment. You need to know the input, the output, and the pathway where this happens. So let's take, for instance, the first pathway we did. If this is empty, you see the carbohydrate in your diet. If that is empty and you have glucose here, what is the process that converts dietary carbohydrates to glucose? What is the pathway or the process?
Carbohydrate digestion.
Carbohydrate digestion, excellent. And then you have glucose. Let's suppose glucose is empty and you have an empty box here or pyruvate here, and they're asking you, what is the process that converts glucose to pyruvate? And you would say it will be what?
Glycolysis.
Glycolysis. And if you get another question that says there is input pyruvate and the output is glucose. And what will be the pathway for that will be gluconeogenesis.
Yes.
A lot of students get this wrong because they don't look where the arrow is pointing to. And so you have to be very careful where the arrows are what pointing, because that determines what is input and what is what output.
Output.
Yes. For instance, let's have a look at this. You have another one, and you have acetyl-CoA and then a cyclic pathway. The name is missing, and they ask you, what is that process? And everyone's clear that acetyl-CoA enter what pathway?
Yes.
Acetyl-CoA enter the citric acid cycle. Is that clear for everyone?
Yes.
Yes.
Okay. Now let's come back to the final pathway, electron transport chain. What is the input for the electron transport chain? Do we still remember that one?
NADH and FAD.
H2. Excellent. And what exactly are they transporting across?
Hydrogen.
Hydrogen electron.
Electrons. Excellent. And what will be the final acceptor for this?
Pardon?
What's the final acceptor for the electrons and hydrogen? What is out here that picks them up?
Oxygen.
Oxygen. Okay. So if you see oxygen is there, and then there's an arrow that points to water, and they're asking you, what is it that oxygen is picking up in order to form water? What will be that?
Hydrogen.
Hydrogen and electrons. Excellent. Now, so carbohydrate is clear because a lot of you wrote your assignment on carbohydrate. So you can see what is input, what's output, what the name of the process, glycolysis, gluconeogenesis, what is the output for glucose, pyruvate, what is the output for gluconeogenesis, glucose. You already know the pathway for acetyl-CoA. Now we come back to the lipids. That's what we did second. And you get a question where there is fatty acids, and input is fatty acids, output is acetyl-CoA, and you've been asked to find out what is the pathway that converts fatty acids to acetyl-CoA, and everyone knows that will be
Beta oxidation.
And a lot of students get it wrong because sometimes they just write lipolysis. Why is that not lipolysis?
Because lipolysis breaks down fatty acids.
It breaks
down
tags. Lipolysis breaks down the tag, which is the lipid dietary 1,2 glycerol and what? Fatty acids. Is that okay there?
Yes.
Yes, okay.
Yep.
And if you have one where the input is acetyl-CoA, especially from carbohydrate and protein amino acids, and the output is fatty acid, what is the name of that pathway?
Lipogenesis.
Lipogenesis. Excellent. So that's straightforward there. And then we come back to the dietary proteins. That's dietary proteins. If you see dietary proteins here, and that is missing. So if this is the input, what will be the output of dietary proteins after digestion?
The amino acids.
Amino acids. And if you have amino acids and you undergo transamination, deamination, and you break it up into two parts, this one will be ammonium ions and what? Carbon skeletons. Everyone's clear on those ones too?
Yeah.
Yes. And if you have ammonium ions and the output is, if this is empty, the output of the urea cycle will be what?
Urea.
Urea. And it's never what? Urine. Some students sometimes mistake urea for urine. Any questions on these pathways? That's 4 marks you can easily get. What's the input? What's the output? What is the pathway, or what's the process? And then finally, make sure you can check where the arrows are what? Pointing to.
Yep.
Any questions?
No.
Caroline, you okay?
Yeah.
Okay. So now we come to the part C. Part C is 7 questions, and I want to be clear. I want to make sure that everyone is clear what to do in part C. Each question of part C, there will be option A or option B. And usually it will be questions that are very similar to what you've already written in your assignment. And so you just choose the one you like. And there are 6 questions for each question. 1, 2, 3, 4, 5, 6. Very simple questions. What is the input for glycolysis? And you just write what you think the inputs are and what is the input used for. Make sure you read a question properly. It asks you what's the input for glycolysis and what's the input for. And you say the input is glucose, you get a mark there. And it's so that it will be broken down to finally generate what? Pyruvate and subsequently ATP. And you get a maximum of 2 points. There are 2 points for each of the 6 questions.
So you have to mention the final product?
Yes, you look at the question and make sure that you answer it carefully. Anytime you put 2 answers there, you're going to get 2 marks.
Yep.
Yes. And it means that some people can get 12 out of that, but the maximum you can get is 8. So what that means is even if you cannot answer some of the questions and you were getting 2 and 2 and 2, you can still get the maximum there. Does that make sense?
Yep.
Okay, so make sure we will look at some of the questions and then how you making sure that you fully understand it. So each question is 8 marks. Each of the points is a maximum of 2. If you just write what is the input for glycolysis and you write glucose, sometimes you get just 0.5. The input is glucose, which is broken down. You can just explain it slightly, broken down to generate pyruvate, so we can get ATP out of it. 2 marks.
Yeah.
Is everyone clear on this one?
Yes.
Okay. And so we have 8 of those questions, and question number 1 could be, because we are starting from enzymes, all those questions that you'll be asked to answer are already being given to you. It's all in your tutorials. And so if you go through the exam guide in the assessment section, you see this.
Yeah, that's what I've been working on.
Okay, that's perfect. If you can go through all of them, it means you can get 100%. So we look at what we would do is, for those who were here the first day, who was here last Wednesday?
Yeah, I was there.
Okay. And so it will not be fair to just go through all of them again, but we will try and start the first couple ones, so others will know exactly what that means. So what this means is you go to, the exams is starting from session 15, and session 15 is the enzymes. And so it's asking you to go back and review the question, session 15 question. What's the question number there? I can't see that.
3.
Okay, so then you go back and, okay, let me go and review that question. So you go into, I'll go to that one. Session 15. And so that's for part C, that's all you do. Question number 3. No, I think I did the wrong one. Session 15. Question number 3. So question 1, it's what is the function of an enzyme? And then because you've done this question before and you also have the tutorial answers as well, you go back and make sure that you can fully understand. And if you say that an enzyme catalyzes a chemical reaction, you get 1 mark. But if you're able to say an enzyme functions to reduce the activation energy of it, you also get 1 mark. And the rate of a chemical reaction increases. So it depends on how you answer that question. So if you look at this and say, oh, there are 2 ways I could answer this question. Everyone knows that any enzyme will reduce the rate of the activation and your energy of the chemical reaction. And your lecturer will add 1 mark. And if you say that it catalyzes the rate of a chemical reaction, or by reducing the activation, it increases the chemical reaction rate, then you get another point. And then question number 2. So it depends on, so let's say you answer that, you get 2 points there, and you cannot answer question number 2. It means that you're already on your way to getting the maximum marks. Question number 2. The cause of an enzyme catalyzed reaction, what actually happens when an enzyme begins to act? And then if you know how an enzyme acts from your notes and also from the tutorials, first the substrate will bind to where? The active site. So the first point is the substrate binds to the enzyme active site. Once you know that, you get 1 point. And then you say that once it binds to the substrate, it forms what? An enzyme substrate complex, and before it releases the product, another point. I hope you are getting the point here.
Yes.
Yeah, so when you are studying these questions, make sure you know all the points there. Don't just write 1 point. Sometimes if it asks you and you just write, you get 1 point, fine. But it means that if you write 1 point, 1 point, 1 point, the maximum you get for that question is what? 6. But you can get maximum of 8, which means you can get all 56. Next question, question number 3. Why is it that each different chemical reaction requires a different enzyme? And you know why enzymes act? Enzymes will only act if the substrate is able to bind to the active site.
Yep.
And the substrates are all different sizes and different shapes. And therefore, most substrates will require specific enzyme in order to bind to it. If you explain it that way, specific shape to fit a specific shape of the active site, 1 point. And only enzymes and substrates that fit together can allow the reaction to be catalyzed. It doesn't have to be the exact words here, as long as you understand what's happening. Everyone's clear on that one too?
Yes.
Yes.
And then question 4. Explain induced fit model. And if, for instance, if that's difficult for you, the induced fit, what that means is that, for instance, if you had different types of proteins, different types of proteins, fish protein or milk protein, and yet the same gastric pepsinogen or pepsin is able to digest it, it means that pepsin sort of is able to, the active site in pepsin can somehow change to fit into milk protein and salmon protein. Does that make sense? And which means that the same active site can change slightly, so individual substrates can fit into the active site. So,
Yep.
And then you have the lock and key. Lock and key means it has to be exactly the same. The substrate has to have that fits exactly into the active site of the enzyme in order for the reaction to take place. Everyone's clear on that one?
Yes.
All right.
Yes.
So the part C, how would you go about studying part C, Caroline?
Sorry, part which?
How would you go about preparing for the part C of this exam?
Good.
I'd continue going through from what I'm doing, the questions from the tutorials, the specific ones that have been said for each pathway
Okay.
and for each thing, and also quizzes, and also
Okay. Yeah. For instance, there's a few questions sometimes that comes up and students struggle a little bit. Because when there's a question on carbohydrate, sometimes they ask you, how would you, if someone cannot metabolize carbohydrate, what would be the outcome of that? If he cannot metabolize carbohydrates, if someone cannot, if he cannot, and then the same question, it's twisted again, and they ask you, what would be the outcome if one, you cannot metabolize carbohydrate, cannot, And 2, why you cannot? Who can see the difference between the two? Well, yeah, if you cannot metabolize carbohydrate. What do we get from carbohydrate?
Your glucose levels would go high.
Yeah, glucose levels will go high, yes. And what else can happen if you cannot metabolize carbohydrate?
Lipogenesis.
Yes, because if you cannot, then the body will have to fall on lipogenesis. And also you can simply say also that you cannot get energy from carbohydrates. And there's one more thing. Carbohydrate gives us pyruvate, and pyruvate gives us what? Oxaloacetate, yes?
So the citric acid cycle cannot start again.
It cannot start again.
Yep.
But there wouldn't be any time when it wouldn't start completely, because the body will start making what? Gluconeogenesis. Yes.
Yes.
Yes. And so there will be a very little amount in there, but definitely it will affect what? Citric acid cycle as well. So you look at the question, and if you have understood the lecture, you can just write these things up. Does that make sense?
Yeah.
You said it again?
What would happen if you cannot metabolize glucose? Well, that's one question, and that's exactly what you cannot get energy out of that. You can either be low what? Oxaloacetate. The body will fall on using what? Lipogenesis. Anytime you write any of this, you get 1 point.
Yep. Okay, yep. Excellent.
And then, yes, and then they could also ask you, why would someone not be able to metabolize glucose?
Is it because the pancreas isn't producing enough insulin, maybe?
Yes, that's that 1 point. If the person has insulin resistance, they cannot. Glucose will never enter the cell. Yes, is everyone clear on that one?
Yes.
Yes. And also, sometimes if you have enzyme, because metabolism is based on enzyme activity. If there is enzyme deficiency or there's mutation in your enzymes, you cannot metabolize it as well.
Yep.
And so that's, you look at the question and say, oh, what happens in this instance, or what will happen in that instance? Everyone's clear on that one?
Yes.
Okay. So we go back to this. We've done most of the carbohydrates. We did the role of enzymes, and then enzyme inhibitors. Who still remember enzyme inhibitors? Session 15, question 4. It's important as well. Pretty straightforward. 15. Question 4. And so we go back to that. And you have to be very clear what you understand with enzyme competitive inhibition and non-competitive inhibition and uncompetitive inhibition. There are 3 of them there. Where does competitive inhibitor bind when they are competing? So where are they competing with the substrate for?
The active
The
site?
The active site.
binding, yeah.
Yes, both compete for the active site, so they bind to the active site. And they will ask you, what about non-competitive inhibitor? Where does non-competitive inhibitor bind?
In the secondary site.
Yeah, regulatory site. And what happens? And then it asks you, where does a non-competitive bind? And what happens once it binds to that regulatory site?
It changes the shape of the active site so that the substrate can't bind with it because it doesn't fit.
So if you can answer it in those 2 ways, then you get 2 marks. What I'm trying to make sure you remember is make sure you answer the question fully in order to get the maximum marks there.
Yep.
Try and get the maximum marks. Because if you get the maximum marks, it means if there's one question you don't fully get, you will still get the maximum full 8 marks there. Is that okay?
Yes.
And what about the uncompetitive or irreversible inhibitor? Where does it bind? And In the active site, and in what way does it bind to it?
Sorry?
In what way? Because it binds to the active site, but not like the competitive. It binds to the active site
Is it with covalent bonds?
using what? A covalent what?
Bond.
A bond. Permanent covalent bond. And therefore, once it binds there, it's permanent. It will never move off.
Okay,
Is that
yep.
clear for everyone? And you can see it all in your tutorial notes. For instance, uncompetitive inhibitor changes the shape. Yes, because it's bound there into the, as a result, substrate cannot bind to the enzyme active site. It's covalently bonded. It's once it's sitting there, the active site is changed and the substrate cannot bind. If you explain it in that context, then you get your full mind. Any questions on these two carbohydrate ones? The next question, let's have a look again. The structure and function of ATP. Who still remember what the ATP structure is? Even if you haven't gone to session 17.
Pardon?
What's this? If you still remember the
Adenophosphate.
ADP.
adenosine and what else is there?
3-phosphate.
3-phosphates.
3-phosphate. There's one more thing.
And plus energy.
And ribose. Yes.
Right. Yeah.
Yeah.
Oh, yeah, the sugar. Yeah.
And the sugar. And then there will be, if they ask you a question, it will be, and where do you get the energy from it? The energy is in what? What's the name of that bond?
Dihydride.
Phosphoanhydride bond. So the best thing is go to the question again. And it says to what question should we go to? It says, let's just a second. It's structure of ATP, session 17, question 3. Is that okay?
Yep.
So you open that up. 17, 21, 17, question 3. Let's go back to that one. Yep, this one. So exactly the same question. You don't have to do much. What are the components that make up the single ATP unit? And you say one ATP, an adenine or nitrogen base. It has a ribose sugar. It has 3 phosphates. And it's linked by a phosphoanhydride bond, depending on what the question says. Does that make sense? So remember these 3 things. Adenine nitrogen base. It's 3 phosphates, 1 sugar, and the bond is important. Remember the name of that bond in that sugar, because that is a bond which, when it's hydrolyzed, releases what? Energy. And when it's formed, you get what? You need energy to what? Form it, as you do in your oxidative phosphorylation. Any questions in that one, in that first part?
No.
Question number 2. In a catabolic reaction, such as glycolysis, is ATP used or produced? What do you get when you break large? You get energy or you use up energy?
You get energy.
You get energy. Anytime you, what about anabolism? When you do anabolism, what happens? Do you use energy or you release energy?
You use energy.
You use energy. So you get those. And so you can see the point there. Catabolism reactions break bigger molecules, if you explain it that way. And anytime you break large molecules, you release making ATP. And then some catabolic reactions produce anabolism. On the other hand, you use energy. Next question, question number 3. Question 4, how much energy is released from ATP when one phosphoanhydride bond is broken? And everyone knows that 7.4. Anytime you break a single phosphoanhydride bond, you release 7.3 what? Kilocalories of energy. Any questions so far? Is Georgia still there?
Yes, yes.
And are you okay?
Yes, all good.
Okay, all good. All right. And so you go step by step, take your time, and go through this step, one step at a time. So if you do the carbohydrates, and then let's look some of it you will have done in your assignment. For instance.
But do these all have to be answered in sentences? Is it okay to sort of do like just?
Well, for instance, if you, this is how the marking guide is. If someone writes, for instance, what's this, if there's a question like, what is the output, input for glycolysis, and what is the output used for? Sometimes a student will just look, what's the output or input for glycolysis? You just glucose, and you get 0.5 mark. And what's the maximum you can get for this thing? 2. So read the question carefully. The question will read something like that. What's the input for glycolysis, and what is the input used for? And then someone will write the input for glycolysis is glucose, and glucose will be broken down into what? 2 pyruvates, which can subsequently be used in the produce acetyl-CoA for energy to go through the citric acid cycle, and the person gets 2 marks. So don't read the whole question. You don't finish halfway and stop. What is the input for glucose? And then at the end of it all, it will ask you what's the input used for. If you just write glucose, you get 0.5.
Next halfway.
Yeah, this means you're halfway through answering that question. Is it okay? And then glycolysis. Glycolysis, questions like, what is the input for glycolysis? What's the input used for? And what is the output for glycolysis? And what would that be used for? And what is the principal events in glycolysis? Everyone still remember a principal event in glycolysis, isn't it?
Pardon?
The principal event.
Principal event in glycolysis means you have to put at least what? Describe what? A reaction there. And a simple reaction in all of this is a redox reaction.
2 ATP, it produces 4.
Yeah, you can see in step 7, it's a substrate level phosphorylation, you generate ATP. Step 10, yes, the substrate level phosphorylation, ADP, a phosphate is added to ADP, and you get ADP. So section 6 is a redox reaction. NAD plus gets reduced to what? NADH. Depending on how you write it, you can get it 2 marks.
Excellent.
Yeah, is that clear for everyone?
Yes.
So which of these, on this, there is another section, the next one. When we come back next week, we'll do that. So today we're going to try and see if we can finish this. What will be the one that you think we should do? Citric acid cycle. Everyone's clear on citric acid cycle? You could easily understand that if you have finished your assignment.
Probably the complexes in the ECT for me.
For you, which one?
In the ECT, if there's anything about the complex.
All right. Now let's have a look at let me see the ETC one and that one. Let's go back to that. TP generation now. Where do you find that question there?
It's under citric acid cycle, under glycolysis.
Okay. Yeah, the electron transport chain, session 20, 1 and 2. Good. Let's go to it. I'll explain it in first, and then when we go to electron 20, let me bring it up. 20, 1 and 2. Question 1. Now, what is the electron transport chain? Describe the location where it is occurs in the mitochondria, by referring to 1 or. Complexes 1 to 4 that make up the electron transport chain are embedded where? In the inner mitochondrial membrane. So electron transport chain takes place where? In the inner membrane of the mitochondria. Does that make sense to everyone?
Yes.
So let's see whether we still follow mitochondria. Mitochondria is the powerhouse of the cell. If you look at it, there is a membrane, outer membrane, and then there is intermembrane space. We are trying to walk from the cytosol into the mitochondria. There is the outer membrane of the mitochondria, and then there is the intermembrane space. There is the inner membrane of the mitochondria before you get inside the mitochondria itself, which is the matrix. And then these complexes are embedded in the inner membrane of the mitochondria. So for ETC, it is electrons that are coming off from NADH and FADH2 that are coming from the citric acid reactions in the matrix and then being passed on. So simply, ETC is a flow of electrons from one complex to the next. But because these complexes are fixed, you will need a mobile electron carrier to shift it from 1 to 3 and from 2 to 3 and then cytochrome C to that. So depending on what question has been asked, you just say that.
Yep.
And then how is the two ETC and oxidative phosphorylation linked? Who still remember that one?
The concentration gradient
Yes.
increases, and this is where I get the spaces mixed up.
Okay. Now, let me explain that so you can be clear about that. You see how anytime you get electrons flow from Newcastle to Sydney, you get your light up. Because the flow of electrons is energy. That's what makes your electric bulb lights up. In your cells, it's the same electrons. Electrons in nature are the same. They are all the same electrons. And so as electrons move from protein to protein, they are generating energy. And that energy is able to pump hydrogen from the matrix into the intermembrane space, hydrogen ion. So the energy of the electron flow is what the proton complexes use to pump the protons into the intermembrane space. Do you get that part?
Yeah, I do. I'm just trying to conceptualize. I always just can't remember the actual spaces and how they are from the outside to the inside.
Okay. So it depends on whether you want to walk from outside of the mitochondria to the inside, or you walk from inside to the outside.
Yep. Okay.
Yeah, if you get it that way, then it's straightforward. If you are inside the mitochondria, is the matrix. And the reactions have taken place there.
Yep.
And so the next level is the inner membrane.
Yep.
The protein complexes are all attached there. And then after the inner membrane is the space between the outer membrane and the inner membrane. That's where the hydrogen ions will have to be moved to. And so as the electrons are going across, energy of it is used to pump the hydrogen ions from the substrates into the intermembrane space.
Okay, yep.
Yeah. And then as they go there, they become so much of it in the intermembrane space, and very little in the matrix. That's what we call a gradient setup. Gradient is you have too much on one side of a membrane and very little on the other side. When you have that, it's just like having a dam behind water behind a dam. It's energy. If you allow it to come back, it would be used to generate more energy. And that energy is what allows that enzyme, ATP synthase, to combine phosphate and ADP together to form ATP.
Yep.
And anytime you break the bond again, energy is released. So let's try and follow this question then. Describe, number B, describe the location of the oxidative phosphorylation in the mitochondria. And because oxidative phosphorylation depends on all of these things, first, the matrix is important because unless the electrons move from the NADH and FADH into the proteins, you won't have the proton being pumped out. The hydrogen is also in the matrix, so the matrix is important. Second, it goes into the intermembrane space, so that's part of the oxidative phosphorylation as well. And then the inner membrane, because that's where the proteins are located. So all of these places you need to mention to get the maximum 2 points. You okay, Georgia?
Yes, all good.
All good. And Caroline?
Yep.
Okay.
Good.
Any questions?
Not at the moment.
Okay. Next question is, what is the starting input for the electron transport chain? Everyone knows this by now, isn't it?
NAD,
Yes, and
NAD,
And FADH2.
ADH2.
and NADH are the starting input. And then if you look at the question carefully, some students will just write NADH and FADH2. But who can read the rest of the question?
final output.
But yes, and what else is there?
The reaction.
You need to mention that it's electron rich.
Why is the final output important?
Oh.
Yes? Okay, the final output of the electron transport chain is what? Water. Yes. And what is the final, if there is no oxygen to bind the electrons and hydrogen to form water, what happens to the electron transport chain?
Pieces.
It will what? Stop.
Yep.
And what happens when the electron transport chain stops? That's also another question.
No energy. No
No,
energy.
no energy, no ATP. And what would, if someone would say there will be no ATP, they will get 1 mark. Someone will go on and said, if there's no ATP, what happens to the cells?
They die.
Yes. And what happens if your cells begin to die? What happens to the organs where they are part of?
They also
They
die.
stop functioning.
They stop functioning. And if your organs begin to fail, what happens to you yourself? This anytime someone dies, they say, oh,
Yeah.
organ failure. The organs begin to fail and you die as well. So depending on how the question has been asked, if you go and say, oh, if the electron transport chain stops, there's no ATP formation, the cells begin to die, the organs begin to fail, and eventually the person dies. It doesn't take any longer to do that, and you get 2 marks. We're just looking at the question, how it's been asked, and following it exactly.
Excellent.
Everyone clear on that one?
Yes.
Okay, now the next question, 1 and 2. Where does the input required for the electron transport chain come from? And you know that the NADH and FADH2, you get some from, mostly from the citric acid cycle.
Yep.
And some from glycolysis as well. So depending on what you say, you can get a 2 mark there. Glycolysis will produce some, citric acid cycle will produce the rest, and you get some from beta oxidation as well. Now, the next question. What makes ETC coupled to oxidate? Why are the 2 of them together? If you cannot get electron transport chain to function, oxidative phosphorylation will also fail.
Mm.
Everyone's clear on that one?
Yep.
Because the flow of electrons is what is required to pump what? The hydrogen. If you cannot flow electrons, you cannot pump hydrogen. If you cannot pump hydrogen, oxidative phosphorylation is dead. Everyone's clear on that one?
Yes.
And
Yes.
so that's why they are linked. The flow of electrons is important for hydrogen ions finding their way into the intermembrane space. And once you have hydrogen ions in the intermembrane space, they will have to flow back according to the gradient. And that's what the energy of it is used by the ATP synthase.
Yep.
Everyone's clear on that one too? Now, who can read the next question for us? The 5.
What is the final output of oxidative phosphorylation? Why is this output important?
A lot of students in the exam condition will just write the final output of oxidative phosphorylation is what? ATP. And how many marks will that person get?
1.
1.
And then the person who reads the whole question, why is the output important? Why do you need ATP?
For your
To
organ.
derive energy.
That's the ATP is the energy. Why do we need energy? What does cells need energy for? That's the question.
Just to survive.
Yes, if you do that, you probably get 0.5. And then it says ATP is used to all the cellular processes, all the cells processes, including chemical reactions, including active transport. to keep cells alive. So if you do that, you get another point, so 2 marks. So don't just read the first question. If there's only one question, then fine. But if there is a supplementary question as well, make sure you answer that as well. Everyone's clear on that one?
Yes.
Yes.
And then there is the second question 1 and 2. Important reactions of the electron transport chain. Important reactions of the and oxidative phosphorylation. What would you be able to do? How would you describe these important reactions? What exactly is that reactions? Redox, yes. As soon as you remember that these are redox reactions. Electrons are stripped from what? NADH and FADH2.
Yep.
By what strips them off? The protein what? Complexes. Yep.
Oh,
Yeah. Are stripped, so you can say that. Electrons are stripped and by the components of the protein complexes, or someone will say this FMN will accept NADH electrons from NADH and FADH2 and become what? Oxidized and pass along the ETC. Just make sure that you follow how these have been answered and do it exactly.
yep.
Any questions?
No.
Okay. All right. What about this question? Describe the component that allows electrons to be passed between complexes. How many of them are there? Someone will say coenzyme Q, probably get 0.5.
There's also cytochrome C.
Cytochrome C. And where did they take it there? Where does coenzyme Q pick up electrons from?
From complex 1 and 2 to complex 3, whereas cytochrome C brings the electrons from complex 3 to complex 4.
Yeah, so if you do it that way, you get a maximum of what? 2. If someone say, just mentioned the mobile electron carrier, you probably get 0.5. And someone says mobile electron carrier and cytochrome C, you get 1. But if you are able to then go and say, because if you look at the question, it's asking you, where do they pass it to? Yeah, if you answer it the way it's been answered here, you get 2 marks. So it's very easy to get more. And so that even if you can't answer a particular question, it doesn't matter. Because if you get 2, 2, 2, you get 8. You only need a few to get 8 marks. You can answer 4 questions well and get all the 8 marks. You can answer all the 6 questions well, but your maximum you can get is what? 8. Now, let's go back to this again. What other question do you want to go to again on this side? Or any particular questions you want to ask?
I'm all good.
You all good? Georgia?
Yep. So far, so good.
So
Well,
good?
Up until next time we meet, anyway, that's enough to keep me going.
Okay. So can I ask questions then?
Yes.
All right. All right, I will ask questions. You see this question here, producing ATP from metabolic pathways. Yep. Session. Yep. Now let's just see if you ask, what ATP do you get from glycolysis? Anyone remember that one?
Pardon?
How many ATPs you get out if you finish glycolysis?
2.
2 ATPs, yes. And then you are asked, apart from the direct ATP, what else do you get from glycolysis?
Pyruvate.
You get pyruvate, and what else? You get additional something else.
Oxaloacetate?
Not glycolysis. You can get oxaloacetate from pyruvate.
You
But
see,
I'm trying to get you to, in terms of energy, I'm trying to get you to remember that you get reduced coenzymes of glycolysis.
NADH.
NADH, yes.
oh, okay. Thanks.
Yes. Yes. And anytime you pass one NADH through the electron transport chain, what's the maximum ATP you get out of it?
2.5.
2.5 ATPs. Excellent. And you get 1 mark for that question. Anytime you pass 1 FADH2 through the electron transport chain, what's the maximum ATP you get out of it?
1.5.
1.5. Everyone should remember that. Because once you know this, then you can be able to work out the ATP, whether it's lipids, or whether it's glucose, or whether it's citric acid cycle, or whether it's glycolysis. Now, once you know that, citric acid cycle, how many NADH do we get out of it?
3.
Excellent. And how many FADH2s?
1.
1 FADH2. So if you still remember that every time you pass any single NADH, you get 2.5. So what happens when you pass 3? You pass 3 through it. It will be 3 times what?
2.5.
Yes, and that will be what? 7.5. Is that okay for everyone?
Yes.
Yes. And anytime you pass 1 FADH2, you get what? 1.5. Yes?
Yes.
Yes. And if you total that, you get 9. But there's one additional ATP or GTP you get from the citric acid cycle. GTP. You add it to it, and what will be the total ATP from the citric acid cycle? 10. Does that make sense?
Yeah.
Yeah.
Yeah, it does.
Yeah. And so you'll be asked simple questions like that. And if that's ATP part. So if you are not sure about producing ATP from metabolic pathways, that one there, you can either send me a message and I can explain it to you. It's important that you can get, if anytime I pass 1 FADH2 through the electron transport chain, I'll get what? 1.5. That's clear. 1 mark. Anytime I get NADH through the electron transport chain, I will harvest 2.5 ATPs. 1 mark. That's just as simple as that, 2 marks. And then if you are asked, how many from pyruvate to acetyl CoA, you get 1 NADH. And if there's 2 pyruvates, you get, within that reaction alone, you get what? 5. So these are extra marks you can get in case you are asked. Any questions from anyone?
No.
All right. So when we meet on Wednesday, we would go through the other half of the hormonal control of glucose metabolism. And that's one question I want to probably, if you go home and you go through that as well, they'll ask you, when there is excess glucose in your blood, what hormone is responsible for controlling that, and what happens? Who still remember that one?
Insurance.
Insulin. And if you say insulin, you get half mark. But what would you say when you say insulin is released from the pancreas? 1 mark. To allow glucose to move from the blood into a
The
cell. Another mark, 2 points. So look at how the question has been asked and answer it accordingly.
Yep.
What happens when your blood glucose level is low blood glucose level?
Glucagon.
Glucagon, and which part of where do you get glucagon from?
This.
From the pancreas. Yeah.
Pancreas.
So if a student says, anytime there is low blood glucose level, the pancreas will release glucagon, 1 mark. And glucagon will what? Allow glucose to what?
Come out of the cell into the blood.
Into the blood, and another mark. So it depends on, and that's a very easy question, the hormonal control. And then it asks you, what reactions does hormone control? What pathways does insulin control? Who still remember that one?
Pardon?
Which pathways, if there is, would insulin, the hormone insulin control? Remember, we have carbohydrate pathways, glycolysis, we have gluconeogenesis, we have glycogenesis, we have glycogenolysis.
Glycolysis, glycogenesis.
Excellent. And then you get 1 more. What about glucagon?
Gluconeogenesis. Glyconeogenesis. Glycogenolysis.
Glycogenolysis. And so it asks you, how does these hormones control these pathways? Anytime insulin upregulates one pathway, it what downregulates what? The other one.
Inhibits, yeah.
Inhibits that. And anytime glucagon upregulates these two pathways, it downregulates what? The opposite pathway. And so if a student is able to say that, they'll get all the marks. What I'm trying to point out is look at a question carefully. And if it's asking you more than 1 point, then make sure you put both points down.
I suppose you can't say too much.
Yeah, and if you do that, you get 2 marks. And when you do that, then if for instance, you are not able to answer all the 6 questions, you still can obtain the maximum amount of marks. Any questions? I think we've done 1 hour now. And so what I will ask you to do is go home, and I mean, you're already home, and go over it.